He is driven by a high sense of purpose behind the steering wheel. He is a member of a team of Ghanaian research scientists preparing a new feed considered to be very rich in protein for livestock. This is particularly good news for commercial operators desirous of adding more flesh to their livestock for high turnover. Dr. Siegfried Obresi comes to the secretary of the CSIR Animal Research Institute of Ghana to monitor the experiment, which leads to the formulation of the feed. The black soldier fly is considered to be of no use to man, much less the house fly, which is actually taken to be nuisance. Ironically, the two insects are of prime interest in the new venture by the animal scientists. The black soldier fly is trapped, but the housefly is left on the loose, but only baited to lay eggs on a sawdust. The eggs, when they metamorphose into larvae, are then mixed with the bran of any of the grains to formulate the new feed for livestock. A better alternative to fish meal or feed formulated with soya bean, not only because the larvae are less expensive, but it saves the farmer the workload associated with soya farms. This is a climate-smart technology, avoiding dependence on plant material.
This is a project that started some 5 years ago with Swiss funds. So collaborating institutions are CABI, Switzerland, CABI, West Africa, CSIR Animal Research Institute, Fish for Africa, INRAB at Benin, and University Naziboni at Burkina Faso. The whole idea is that we see that agriculture is now challenged with climate change, consumer preferences, reduction in land size, and production of a lot of wastes. So to be sustainable, there should be a way of reducing dependence on good materials and moving on to things that human beings may not even use, but the animals can use.
3 components guarantees the qualitative nature of the larvae feed.
This is the incubation bowl. You can see the eggs. So we drop them onto the incubator, and then we incubate it in this trough for 5 days. Then they will hatch into larvae. Then we put them into bigger troughs and then do the maturation here. They will be here for about 8 further days. Then they will mature here pre-pupae. It means we don't allow the larvae to pupate, because when they pupate, they will go to adult. At that time, the protein level will change. It will become more fibrous. The chitin will increase, so it will not be very good. So we harvest the larvae pre-pupae.
The scientists' defense of larvae feed is not only situated in science, but folklore as well.
We have a history from the Bible. John the Baptist was eating locusts, which is an insect, and honey, and he survived. And even in Ghana, we are eating the kokono. And if you go to other places, the larvae from shea nut palms are also eaten. So we have a basis to suggest that we should use larvae for livestock production.
You get a clearer picture as to why the scientists would like to take further steps to improve on current conditions when you obtain insights at the Animal Feed Quality Control Laboratory. The side effects of other protein sources, such as excessive soya bean and fish meal in animal diet, places the larvae feed on another pedestal.
The soya bean source has its effect when it's given in excess, just like the plants, the animal source too, which is the fish meal, which is normally brought. If soya bean is fed in excess without concentrating on the recommended values, for example, poultry, they can have pasting up, or they have excreta sticking at the vent, and so they will have finally get renal dysfunctioning. So it's an example of improper feeding. Just giving them anything because you think the protein is there, and so you just feed them. So it's an effect. Another example is the fish meal. If it's fed in excess, you can have fishy flavor in your meat products or even the eggs. The flavor wouldn't be so attractive or nice for consumers.
In analyzing animal feed in this lab, it is to ensure animals are on balanced diets, free from contaminants and toxins.
An example of feeding fat supplemented diet without really knowing what the amount should be, just giving them, is that if you feed early cows, lactating cows, with fat supplemented diet, they may end up having ketones in their bloodstreams, and so they'll have a condition known as ketosis.
High moisture content of animal feed reduces the amounts of nutrients in the feed, just as the parameter is a warning that a feed may have short shelf life in storage. The CSIR Animal Research Institute of Ghana provides additional lab services for parasitology, biochemistry, microbiology, and biotechnology. We will go through one by one, and for starters, it must be stated that the parasitology lab performs diagnostic tests in both animals and humans to determine whether there are transfers of disease between the two. It is reported that the animal that is disinfected becomes predisposed to another infection if the animal released into free range comes into contact with other animals. The two-way transmission mode between animals and humans is startling.
One of the things I found out was that the farmers are also infected with some of the parasites that are in the animals. And we also found some goats that were infected with species of parasites that are supposed to be in humans, which means that there is transmission between the animals and the humans. And one of my aims is to do more research to find out how these transmissions come about, how the infections go about, and to come up with preventive and control measures. Yeah.
A very important tip for those rearing livestock to beware of zoonotic diseases. The biochemistry lab performs a wide variety of functions. It takes urine, fecal, and blood samples of animals for analysis. These are to determine the state of animals' vital organs and hormones.
The biochemistry, we carry out samples from the scientists, from the universities, from outside. And they bring us the sample, we analyze, we do full chemistry. We check the liver function test, that's what we call the liver profile. We do the kidney function test with BE creatinine. We also check hormones. Then, as far as the chemistry is concerned, usually the point they bring is the serum we use or plasma. When they bring the blood sample, then we will separate and use it to run the test.
In biotechnology, the lab develops technologies to address challenges in livestock industry. Such technologies are meant to transform lives of farmers and value chain operators. It devised a system which has made a major breakthrough in the breeding of Guinea fowls.
One of the challenges of Guinea fowl breeding is farmers are not able to differentiate Guinea fowls, a male from a female, so that they can embark on breeding program. So in this lab, we developed a technology based on polymerase chain reaction to differentiate male Guinea fowls from female Guinea fowls right from day one.
When all indicators have failed to reveal the exact causes, the scientists fall on genetic characterization as the last resort, and this is performed by the molecular lab.
Molecular biology works with DNA or RNA, and there are genetic materials. Whatever the phenotypic characteristic which come out, they're all ultimately controlled by our DNA or RNA. So therefore, the ultimate diagnosis or characterization lies with our genetic material. Yeah. So genetic characterization, DNA-related or RNA-related information gives us a very fine differentiation. Although two, say, pathogens, they look the same, but at a very fine level, we can differentiate using molecular markers, etc.
Microbiologists at the CSIR Animal Research Institute indicate that worldwide, 80% of diseases are caused by microorganisms, especially bacteria, seen only in microscopes.
Microbiology laboratory of the Institute deals basically with routine diagnosis and research in the areas of bacteriology, mycology, virology, and immunology or serology. And when it comes to routine diagnosis, we deal mostly with animal farmers in the handling of their poultry, sheep, goat, cattle diseases. Sometimes they bring a dead animal for post-mortem, and then we take the organs for culture and sensitivity to determine appropriately the drug of choice for treatment.
Mortality in animals induced by wrong cultural practices or negligence is real. A veterinary surgeon of the CSIR Animal Research Institute is pictured here, performing a post-mortem on 2 bears. The cases were reported by a farmer.
A farmer brought turkey, 1 turkey and 1 broiler. And in my post-mortem results, I found out the broiler had a necrotic enteritis, which is an intestinal bacterial infection. And the broiler had some form of paralysis, which I'm still talking with them, getting more clinical history to do the final diagnosis. I receive quite a substantial day where I do PM to tell them the cause of the death of the animals and also offer solutions in terms of medical treatments that they should follow. For companion animals, I provide services. Animal Research Veterinary Hospital provides services in surgery if you want to do some form of castration for your male dog, or you want to spay your female dog, or you want any medical assistance in terms of commonly dog diseases and cat diseases, or any companion animal, you are welcome.
The farmer will have to put them on.
Based on the results of the post-mortem, the farmer may be advised to bring other bears which were in close proximity with disease-stricken ones for tests, or unaffected ones quarantined to avoid the spread of disease. In Ghana, small and large ruminants transported from the north to the south of the country begin to show symptoms of a disease called PPR. It is marked by greenish diarrhea as well as purulent discharges from the animal's nose, eyes, and mouth. Having demonstrated world-class capacity to deal with animal diseases, the Rome-based Food and Agriculture Organization of the UN declared the CSIR Animal Research Institute of Ghana as West Africa's Animal Disease Treatment Center. This was an international license granting the institute the right to provide veterinary services in an entire sub-region. We make reference to the beginning of the script as we introduce the Quail. It is an emerging poultry line being handled by the CSIR Animal Research Institute at its field station at Fafraha, near Accra. The Japanese species of the quail in its custody were fed with the insect larvae to improve on the bird's reproductive performance.
Between 5 weeks and 6 weeks, they start to lay their eggs. And when you compare in terms of real economics, the quail takes in about 18 grams of feed a day, whilst the laying hen takes 115 grams. But when you get to the market, the prices of the eggs are fairly the same.
The poultry profile of the CSIR Animal Research Institute of Ghana features broilers raised for their meat, layers for their eggs, and multiplication, and the Aribro, which is a quality variant developed by the Institute. Director of the Institute is Dr. Ebenezer Ansa.
We have a hatchery that can produce day-old chicks, and it has a capacity of about 30,000 day-old chicks, which we have on offer on commercial basis. If any farmer wants to bring his eggs to hatch them, we are prepared to do that for them.
One area of which we are so much concerned is the high importation of day-old chicks. Currently, all the day-old chicks that are being housed or being managed by our poultry farmers are sometimes imported. We stand a chance. We have a hatchery capacity of close to about 30,000. So we do not see the essence of trying to import day-old chicks from Australia as far back. You can imagine when you are importing day-old chicks from Australia. Australia is far off. By the time you bring day-old chicks from Australia to Ghana and then moving them from here to, say, Afara farms in the Brown region, you can imagine the stress on them. So we are now in a better position to produce day-old chicks that can be enough to feed the country Ghana.
Drugs used on livestock are transferable to human consumers. Thus, the Institute performs what it calls drug withdrawal before birds are slaughtered and dressed for sale.
We don't normally give them a lot of drugs because we want to prevent drug resistance in humans when they consume the birds. And before we slaughter our birds, we do withdrawal of drugs 2 weeks prior to slaughter so that we don't have any residuals in the dressed meats.
New technologies designed by the CSIR Animal Research Institute would mean that there is labor efficiency in piggery and quicker growth in pigs, as the monogastric animal will be ready to go in a maximum of 5 months. Making all the difference is the laying of sawdust in the modern pig sty.
The animals on the sawdust normally do not come out with this stench that is associated with piggery. The animals on this technology also grow faster as compared to those on the conventional type. Again, you do not need a lot of water to wash the floor and again to wash the animals as we normally do on the conventional type. You just need cool drinking water for the animals to just drink and they are fine and free to go. You can also construct this particular structure, which is quite cheap as compared to that of the conventional type. Again, if you can look at the type of structure that we have, you can see that we have the translucent roofing sheet, and this roofing sheet helps in directing sunlight straight onto the sawdust. Now, there is a solution that we may talk about maybe in the course of the presentation. The solution normally, when sprinkled on the sawdust, converts the sawdust into simple mushrooms and sugars that enables the animal to eat as supplements. In view of this, it enables the animal to grow faster as compared to the conventional type. Again, at the end of the production phase, the sawdust is not just a waste, but you can scoop this out and it will serve as manure for those who are interested in cropping vegetables.
Oti region is reported to be a hub of small ruminants in Ghana. In other words, the eastern corridor of the country has the highest density of such animals. Working in close partnership with the Animal Production Division of the Ministry of Food and Agriculture, the Nkwanta Livestock Breeding Station of the CSIR Animal Research Institute is into protocols to multiply sheep and goats.
Apart from the Nkwanta breeding station here, there's no other research station in the area to address issues in livestock production. Talk about nutrition, talk about livestock production as a business, talk about health issues. And so to be able to advance the kind of help that farmers require in livestock production, the Animal Research Institute is teaming up with MOFA APD to transfer a new breed of sheep that Animal Research Institute has developed to the area so we can multiply and transfer the offspring to farmers to improve their own stock so they can produce to improve their food security and also to improve their income.
The institute has another field station for ruminants at Pokuase near Accra.
These particular ones, you can see that they are with their young ones. So we don't call them mother, we call them dams. Then with their lambs. So we don't allow, as soon as they are due for parturition, we separate them from the main flock into the maternity. So this area is called maternity unit. Maternity unit. So we bring all of them until the time that we wean them at certain weight or an age. Then we separate them. And then we put the male on them again. So we don't allow the females to rest for a long period.
In cattle breeding, the CSIR Animal Research Institute has been crossing different breeds to attain optimal results in meat and milk production. By the estimation of the scientists, Ghana has a shortfall in meat consumption, thus production is subsistent to meet local demand. The Food and Agriculture Organization FAO pegs daily protein requirement in people at 70 to 80 grams, of which 50% must come from animal origin. The average meat consumption by Ghanaians is 46.5 grams, below the FAO standards. Again, Ghana imports 60% of meat to supplement domestic meat production. The CSIR Animal Research Institute reckons that the void can be filled should there be private sector partnerships and additional funds to intensify research for multiplication.
We've been able to develop new breeds of sheep, goats, and cattle, which are bigger. And what that means is that they are more meaty, and they are also resistant to diseases compared to the local ones that we have. So we have a kind of hybrid between two different species that have a plus and also a minus, and we've been able to combine both the pluses from either side whilst we eliminate the minus that they have.
In 2009, Argentina, the South American nation better known for beef production, invited Ghana to discuss ways of cooperation in animal husbandry and beef processing. Should that happen, the focus is likely to be on this breed of cattle already in Ghana called Indama, reared for its beef only. They are kept on the Institute's fields at Pokuase with other breeds in the head office compound at Fafraha, both locations on outskirts of Accra. The issue of nomadic herdsmen has often provoked outcry among rural farmers. They say cattle destroy their farms. The animal scientist of CSIR considers Afram Plains in Eastern Region as the best location for cattle grazing.
We've had challenges with the Fulani herdsmen and the crop farmers because during the dry season we don't have enough herbage. So they move their cattle into people's farms, and there's been challenges, especially in the Eastern region. Now we're doing work to address this issue. We've been part of the national committee to see to addressing this issue. Part of the work we're doing in the Afram Plains and trying to see if we could develop pastures so that we get these cattle farmers to move their cattle into those ranches. We are currently trying to see if we could develop corridors because we have animals moving from our neighboring countries, Burkina Faso, Togo, Cote d'Ivoire during the dry season into our countries. But there are no defined corridors for them so that they could graze and they could water. And currently we're developing those systems.
In the Dairy Product Research and Development Unit, simply called the Processing Unit, cattle products are processed into several items for consumption.
Okay, yeah. So this is our final finished product. This is 1 liter, the volume that is vanilla flavored. And we have the same volume, 1 liter. This is strawberry flavored. This is 300 ml vanilla flavored, and the same to the strawberry.
In meat science and food safety, high protocol standards are observed. It is to ensure that the slaughtered and dressed meat meet best hygienic standards. First, optical conditions at the Institute's abattoir at Fafraha speaks for itself with regards to hygiene, and this is open to the public at a small fee. Those who burn car ties to remove fur on animal carcass are putting lives of consumers at risk. Scientists say the soot being the black, powdery or flaky substance is carcinogenous, meaning it causes cancer. The CSIR Animal Research Institute has developed a technology to avoid that, which is the use of gas to do the singeing. It is at a minimal cost in terms of energy usage if meat to be processed is placed in the chamber of a solar dryer developed by the CSIR scientists. The hot wood technology also acts in place of the car ties by applying gas to cook the meat. Later, a little smoke is introduced on the meat just for flavor and appearance.
We have meat processing equipment here like bandsaw, like the mensa, like the bull chopper, which are available to the public for use at a very small fees. We also provide training in processing, in meat handling, meat safety. Our abattoir, our slaughter unit is also available to the public for use at a small fee.
At the Institute's field station at Pukiase is the grass cutter breeding, where focal persons offer deep insights into its management.
Benin came to learn grass cutter rearing from Ghana. As we talk, Benin, one farmer can put about 3,000, 5,000 grass cutters. But Ghana, the maximum that you can get about 500. Why? Because when the NGOs who came to promote the grass cutter rearing withdrew their services, the promotion exercise went down, and now we've started. We are now, the animal research is now organizing training programs for farmers.
It is an important source of bushmeat for Africans in the sub-Saharan area. Breeding of grass cutter will require mating the opposite sex, which the scientists have found a way around the complicated means.
The grass cutter is in such a way that if you bring the male during mating, if you directly bring the male to the female, they fight till one dies. So how do we resolve that issue? We have to research into it, observe them for some time. So we designed a special cage. Before we made them, the male is brought closer to the female. They will be there for a week, 2 or 3, so that they establish some kind of a relationship. So when you bring them together, that aspect of fighting is resolved. Smoothly they mate.
There are 3 indicators by which to differentiate between the male and the female grass cutter, which was previously unknown.
If you look at the male grass cutter, the head has U shape. It's a gentle slope. But if you take the female grass cutter, it has V shape, a very steepy. So with looking at the head, you can really identify the male from the female. Another thing that we also realized is that if you have the female grass cutter and you want to know, just pass your finger or your palm at the flanks of the abdomen. You'll find some pimples-like structure over there. That is the teeth. So even if the animal is young, you'll be able to see that it's female, or maybe because the male don't have the teeth at all. So this has also helped the farmers to distinguish between the female from the male easily.
Grass cutter meat is a source of protein and incomes, if professionally handled. Rabbitry is the other husbandry undertaken by the CSIR Animal Research Institute. Rabbit meat is said to be high in protein, low in sodium and cholesterol, and it is a good income supplement. Gestation period of rabbits is between 30 and 32 days, and here are the on-station research being conducted on the small mammals. the scientists take a lot of interest in the rabbit's fur or coney for leather. Rabbit hide is used in making hats and clothing and could be a source of export revenue for the self-employed.
Based on the farmer's perceived preference, what the rabbits like, a little research that we did to look at which ones the rabbits like better, we selected 4 plants out of the so many plants, harvested them, dried them, and then formulated diet for the rabbits. And then with this formulation, we incorporated even corn cob. When people harvest the corn, they throw the cobs away. We milled them and then incorporated them into the diet because we want to reduce feed costs.
We have been switching between Pukwase and Fafraha, and we return to Pukwase to see the Pasture Museum. It is the only pasture museum in West Africa with unique grass propagated for multiplication. However, it faces one threat, that of encroachers, particularly real estate developers in the Pukwase neighborhood, who are inching closer and closer towards usurpation of lands occupied by this foliage. From the animal scientists who have taken the pains to observe and code trends in plant behavior, it is reported that at the onset of rains, grasses contain 10% protein, and this drops to 3% in the lean season. It is reason scientists have devised reliable means of formulating animal feed for the optimal intake of protein for their growth.
There are materials that have been researched on issues of biomass yield, issues of nutritional quality have all been assessed, and issues of animal acceptability. These are materials that animals prefer over other materials. So they are not just weeds, as a lot of people refer to. They are pasture materials that animals really eat. But among all the materials we work on, we have kept samples here. So this is a repository of all the materials we keep, the germplasm. We have fields of 4 meters by 4 meters. So we have samples of all the materials that we work on. We have other areas where we do multiplication for wider applications. But here we keep small samples so that when anything happens, if we lose all the other materials on the field, this is the only backup we have. So if anything happens to the materials here, it is a real disaster for the whole country and for the whole West African sub-region.
These are the multi-nutritional feed blocks, whose alternative is pelletized feed. Materials are held together by a binder, and this kind of feed is said to be good for monogastrics, as pigs and poultry. Livestock which are monogastric are animals that have simple single-chambered stomach, compared with ruminants like cow, goat, or sheep, which have 4-chambered complex stomachs. At this point, let's revisit the grass cutter breeder and listen to how the pelletized feed, for example, is performing miracle on domesticated grass cutter.
Grass cutter in the bush, when they conceive, they reabsorb some of the fetuses. What happens is that they see that they don't have enough nutrients that can cater for about 8, 10, or 11 fetuses that they have developed. So they have to reabsorb some, they take some of the fetuses in, so that when they give birth to, say, 5 or 4, they can cater for that. They can have enough breast milk to feed those ones, yes. So then we have to solve that issue. So we have to develop special feed, which we give to the pregnant doe, so that it's able to have enough nutrients in the system, so that the issue of reabsorbing about 40% of its fetuses will be resolved. When we did that, yeah, it has worked. Now, we can have grass cutter giving birth to about 8, 10, or 11 young ones.
The director of the CSIR Animal Research Institute, Dr. Ebenezer Ansah, says the institute is a center of excellence committed to entrepreneurship demands of individuals, organizations, and government.
We conduct cutting-edge research and provide state-of-the-art technologies to industry. The institute consists of 6 technical divisions and 2 non-technical divisions. And we have a wide range of expertise. We have parasitologists, we have microbiologists, biochemists, we have expertise in feed production and nutrition, pasture management, agroeconomics, breeding and genomics, veterinary science, meat science, and food safety. So it's a highly specialized area of expertise.
The Director General of the Council for Scientific and Industrial Research, CSIR Ghana, Professor Victor Kwame Ajimai, says the value chain of running a poultry farm, for instance, opens many doors.
If you take the poultry value chain, for one poultry farmer, you have not less than about 15 jobs that are created within the value chain for that single poultry farmer. Transportation, when it comes to the eggs, transportation, feed, it comes to the vaccines that you have at the farm level, those who plant the maize. So the fact is that in looking at a technology, we do not look at the value chain of the technology and the jobs that are created. And that is one of the things that CSIR is doing, so that you recognize these jobs at each of the value chains, and then you direct people to those jobs. Because people will see, oh, I want to be a poultry farmer, without knowing that there are about 15 other jobs that you can do to get to that poultry farmer.
His reflections are proven by a female poultry farmer who received training at the CSIR Animal Research Institute.
I started poultry about 5 years ago. My intention was to raise layers for eggs, table eggs. But when I started, because of lack of knowledge, I stopped on the way. So 2 years ago, there was a project supported by CAFASI, that's the Korean government, through Animal Research Institute. So I was lucky to be one of the beneficiaries. So in starting that project, they took us, the Animal Research Institute called us for training how to give vaccinations to the birds, feed preparation, records keeping, and how to expand our business. So there were several workshops being organized by the Animal Research Institute, in which I attended. So the first project, the Animal Research Institute raised birds and distributed it to we, the farmers. So upon that, I decided to expand with the little knowledge I got from them, to expand the business to a different level. So in the second year, we were lucky they supported us once again with another base, in which gave us more room to expand. So we decided to expand, I decided to expand from 200 birds to about almost 1,000 birds. That's the second year to come.
Out of the poultry, she has a crop farm fostered by manure obtained from poultry droppings. And now, the MAG focal person of Animal Research Institute of Ghana.
MAG is modernizing agriculture in Ghana, and it's a Canadian-funded project. It's a Canadian-funded project. And basically, one of their main objectives is to do technology transfer. And so they have resourced some CSIR institutions to do this technology transfer on their behalf. Animal Research Institute happens to be one of them. And for starters, we were asked to produce manuals. And so we were able to produce some training manuals from some of our technologies that we had worked on. And so we have a manual here on training training manual on preparation of multi-nutritional feed block for ruminants, which we prepared. There's another one which we prepared, common feeds for ruminant livestock production in Ghana. And then there's a third one, indigenous microorganisms, IMO, technology for sustainable pig production. All these manuals were produced with the help of MOFA, Ministry of Food and Agriculture, the Directorate of Agricultural Extension Services. And after preparing the manuals, what we did was to go around having what we call training of trainers workshops